video is going to take us through Lab 7, which is about protein synthesis. To start us out in the lab manual, we're just going to kind of talk about some basic stuff related to DNA and RNA. Both DNA and RNA, if you remember from Chapter 2, the chemistry chapter, they are nucleic acids, which means that they are made out of nucleotides. We went over this in lecture when we talked about monomers and polymers. So the monomer, the nucleotide that you can see in this picture, they get strung together to make nucleic acids, which are the DNA and RNA that you guys are familiar with. The nucleotide monomer that's going to be making up DNA RNA contains three components. We have a phosphate group, a 5-carbon sugar, which for DNA is deoxyribose. For RNA, the sugar is ribose. That's one of the differences between DNA and RNA. They have different sugars. And then there's 5 different nitrogenous bases. It lists 4 here, but RNA has a 5th one that we'll talk about a little bit later on. So this is what DNA and RNA basically is going to be made out of. Now when you string together nucleotides, you get what's known as a polynucleotide. So a strand of RNA is a polynucleotide and one strand of DNA would be a polynucleotide. This is how they end up getting strung together. Now without getting things to be super complicated at this point, both DNA and RNA are directional. The directionality comes from this 5' and 3' numbers that they're telling you there, but since you guys are non-majors, we're not going to dive too deeply into that. What I want to point out is this is one nucleotide, this is another. So there are 4 nucleotides in this strand. They've gone ahead and even labeled the different nitrogenous bases. So this is the one that makes this whole nucleotide an adenine. This is cytosine, this is guanine, and this is thymine. These are the 4 possible nucleotides that you can get into DNA. And so this is what a polynucleotide of those 4 would end up getting put together as. Next. When we're going to make a whole DNA molecule, it's actually 2 polynucleotides that are hydrogen bonded to each other. So this is a polynucleotide, this is a polynucleotide. When you stretch them both together and you bond them together, incidentally with hydrogen bonds, that's how you get DNA. The double helix is 2 strands put together. When you put the 2 strands to each other, it's the nitrogenous bases that are going to be facing each other. And there's certain combinations of letters that you can get. So adenine will what's known as complementary base pair with thymine, so A goes to T, and guanine can complementary base pair with cytosine, so G goes to C. G can't go to T and G can't go to A, it can only go to a C. So essentially what that means is if we give you one strand of DNA, you should be able to tell us what the opposing strand is going to be based on those rules of complementary base pairing. Now while the 2 strands are held together with hydrogen bonds, the nucleotides that make up one polynucleotide, these are covalently bonded together. So a polynucleotide is actually very strong, but the 2 being held together, they're held together with the weakest kind of bond we have, the hydrogen bond. So it's actually really easy to separate the 2 DNA strands from each other. Just a little bit of heat will actually do that. Now, I mentioned the complementary base pairs a second ago, but you want to make sure that you get it and you understand it. So again, A to T, C to G, that's how complementary base pairing is going to work. In the lab manual, which I finally have new PDFs for the lab manual, the page numbers aren't set just yet as I'm making this PowerPoint, but towards the middle of this, they give you a strand of DNA, A, G, C, T, and what they want you to do is write down what the complementary strands would be on the opposing strands. So, if there's an A on one strand of DNA, since A goes to T, you know the other strand is going to have a T that is base paired to that. G goes to C, so C goes to G, and T goes to A. That's how complementary base pairing works. So we give you one strand, you just use the opposite letter, and you can figure out what the opposing strand is going to be. Next. Your cells contain about 20,000 different genes that we are aware of at this point. There's probably more than that, but that's what we've been given as data from the Human Genome Project. Each chromosome that you have contains about 1,000-ish genes on it. Some of the longer ones have more, some of the shorter ones have less, but about 1,000 genes per chromosome. What that means is a gene is just one little segment on the whole entire molecule of DNA that builds a particular chromosome. So as we're looking at this picture, this whole thing is a molecule of DNA, but a gene is just one segment of that DNA molecule. And what that segment does is it tells your body how to make something specific. we're going to say that what we're going to be making is a protein because most of the genes that we know about, those 20,000 genes, they're protein coding genes. Genes code for more than just proteins, but at least that's going to get us started on that. Next, me and your lab manual disagree about the way that this is written. I'm hoping they're going to change that in the editing process, but it says that codons are found on DNA, and they're not. Codons are found on messenger RNA. However, a molecule of DNA does get read 3 letters at a time, and so what we say instead of calling that a codon is it's a triplet. So every set of 3 letters that you can find on DNA is called a triplet. So ATG is a triplet, CTA is a triplet. Those triplets are going to be transcribed into messenger RNA, and then once we get it to messenger RNA, at that point we switch names from triplet to a codon. And so that's a disagreement that I have with your lab manual, but there you go. We still do write things in sets of 3 letters, it's just that we don't call them codons on DNA. Next, we're going to get into how the genes actually eventually get us over to a protein, and there's really just 2 steps. The analogy that I usually give students here is I want you to think of a gene like it's a recipe for how to make something. A couple days ago I made some lemon bars. The recipe that I used to make the lemon bars, I've been using that same recipe for a very long time. I don't like to leave it next to the stove as I'm cooking because I tend to sprinkle stuff like powdered sugar or I get butter or egg on it or something, and so it's very common for me to make a copy of that recipe. Well, your cells do the same thing. A gene is a recipe, but you want to protect your genes, and so very often your cells make copies of those genes, and that process of making a copy is called transcription. So transcription just copies a recipe so that you have a usable copy, that it's okay if you spill on it. You still have the original, and you can still make copies of that original if you need to. The gene recipe is written in DNA. The copy that we're going to make it into is called messenger RNA. So the process of transcription reads DNA and makes messenger RNA so that we have that usable copy that we can use. And so on the picture up over here, whenever we have our DNA, remember that the gene is only a part of that. And so we would say the gene is this segment of DNA. It's not even on the opposing strand. Whenever we do transcription, we only copy one strand, the strand that has our gene. The process that gets us from the DNA that we want to copy into a usable copy of a recipe is transcription. And so we just go from DNA to RNA. It's really that simple. Next, since I'm using recipes as an example, translation is the act of reading the recipe and doing the cooking. If you think back to when we were talking about the different parts of a cell, we talked about ribosomes. Ribosomes are responsible for protein synthesis, and here we are talking about protein synthesis. And so ribosomes can be analogous to the chef. What they do is they read the recipe and they cook based on that recipe. There are going to be some things that help us or help the chef actually do their job, but essentially what it means is we read the copy of a recipe and then we cook the dish, and the dish that we're trying to make is a protein, which is made out of amino acids, which are these things listed off down there. So translation is just cooking, essentially. So going into those in a little bit more detail. Transcription gets us from DNA to RNA, or again, it copies our recipe. Whenever we give you a stretch of DNA, you should be able to transcribe that into RNA. It works a lot like the complementary base pairing in DNA, except RNA does not use T's. So any place where you would have wanted to put a T, you would put a U there for uracil instead. Here's what I mean by that. If this were a strand of DNA and we were replicating it to another strand of DNA, this A would go to a T. But we can't put T's in RNA, and so instead you would replace the T with a U. A goes to U now. G still goes to C. C still goes to G. That doesn't change. And T wants to go to A. You can still put A's in RNA, and so that part's just fine. So the only thing that changes is this, and it changes because you just can't put T's in RNA. Next, your lab manual has some practice things to run you through at this point. Oh no, and I have a typo already. I'm going to, what's next to S on the keyboard? Really, none of the letters that actually matter except for the A. I'm going to say that this was probably supposed to be a C. Maybe I should go out and actually look to see what that letter was supposed to be, but we'll just say it was supposed to be a C. Maybe autocorrect decided to correct that for me. Who knows? Anyway, given the following strand of DNA, right, the complementary strand of DNA. So what they're wanting us to do is that first thing that we had done. So A goes to T, T goes to A, C goes to G, G goes to C, A to T. Hopefully that is a C in your lab manual. If it's not, I am very sorry, but C goes to G, then A, G, C, T, G, T. That process that you guys just completed, it's called DNA replication. That process happens anytime you need to make a new cell. So anytime they ask you to replicate a strand of DNA, it means you're going DNA to DNA. So you can put T's in DNA. A couple more questions after that. Given the following strand of DNA, write the complementary strand of RNA. So now, since we're going from DNA to RNA, we're carrying out transcription, which is the answer to number 4, but I'll come back to that in a second. So here's what you need to remember. Anytime you would have wanted to put a T, you can't do that. T can still go to A, that's fine. C still goes to G, G still goes to C, T still goes to A. A wants to go to T but can't, because you cannot put T's in RNA. So this is where we're going to substitute in that U, and then A. Can't put a T. See how you wanted to do that? That's going to be a U instead. G, C, U, A, C. That process of going from DNA to RNA, it's called transcription. So anytime they ask you to transcribe something, it means that you're going from DNA to RNA. So replication, DNA to DNA. Transcription, DNA to RNA. How are DNA and RNA different from each other? So the one thing that I mentioned already was DNA uses the sugar deoxyribose, RNA uses the sugar ribose. So they have a different sugar that makes up their nucleotides. Next, DNA is double-stranded. RNA is usually single-stranded. You can find some examples where it's double-stranded, but most of the time it's just going to be one strand. DNA uses thymine versus RNA uses uracil. They both use cytosine, guanine, and adenine, though. There's also very often a locational difference in eukaryotic cells, so we always keep our DNA inside the nucleus, versus RNA can leave the nucleus and it can float around out in the cytoplasm. You can find some in the nucleus, but it's not constrained to that place. So that's all the ways that it's different. How are they the same? They're both made out of nucleotides. They both have phosphate groups in them. They both use those guys that I mentioned just a second ago. And they are both critically important for protein synthesis. Next. There's a few different kinds of RNA. We're only going to be talking about 3 of them. mRNA is the usable copy of a recipe that we make during transcription. mRNA is what actually contains the codon. So again, your lab manual said that codons are on DNA, but it's not. Codons are on messenger RNA. tRNA is what I like to call the sous chef, or the chef's assistant. What it does is it brings the ingredients to the chef. The ingredients for making proteins are amino acids. So that means tRNA is going to carry amino acids on one side, and then it has to bring the right amino acid over. So it needs something to match up to a codon. It contains anticodons on the other side. Okay, so this is a picture of tRNA. This is one of the amino acids. There's 20 different amino acids. Let's just say that's lysine, just for fun. This is the anticodon right here. The anticodon matches up to a specific codon that you would find on mRNA, and that's how you would know which of the amino acids to bring to the ribosome so that we cooked our dish with the right ingredients. And so it's kind of like puzzle piece shapes. If the puzzle piece doesn't match, that's not the ingredient that we wanted. But if the shapes do end up matching up, then we can go ahead and bring that dish or ingredient over. Next, our RNA is ribosomal RNA, and that's actually part of the ribosome. The ribosomes are made out of protein and our RNA, and so really it's just part of the chef. All right. Next up, kind of coming back to the lab manual, one of the things that you need to be able to do is figure out how anticodons match up to codons. They still use the rules of complementary base pairing, only now we're doing RNA to RNA. So once again, can't put U's in place, so U base pairs with A. So AUG, if that's the codon, the anticodon would be UAC. If CGC were the codon, then GCG would be the anticodon. And if GAC were the codon, then CUG would be the anticodon. So this would be on our recipe. This would be on the chef's assistant, so we bring the right ingredient to the chef. Next, translation is where we again do the cooking. We read the mRNA, we bring the amino acids, and we cook the protein made out of those amino acids. Now the cells do this using that codon to anticodon binding. We have to essentially read a translation book to help us figure out which ingredient gets coded for by a particular codon. Now in the lab manual, they give you this version of the genetic code. You can also find some tables that are out there. But the way that you would do it, I'm going to come back for just a second. We're going to look up this codon in that genetic code, or what they called codon chart in your lab manual, and see what it would code for. So to do it, you start at the center of the circle, A, then you move out to the next letter, U, and then you move out to the next letter, G, and then written off to the side, it would tell you what amino acid that coded for. AUG happens to code for MET, which is methionine, so that's the ingredient that would be brought to the chef for that particular codon. Now we're going to practice. Use the codon chart, aka the genetic code, to write the corresponding amino acids in the blank. So we just did AUG. AUG codes for MET, and so you would put MET in that first blank. Next, AAA codes for lysine. AGC codes for serine. CUC codes for leucine. UUU codes for phenylalanine. And GUA codes for valine. You don't have to know what the three letters stand for, like I kept saying the full name of the amino acid. You guys can just say VAL, and I'm perfectly happy with VAL. If you were majors, I would make you learn what those abbreviations stood for, but you're not. And so that's all you have to be able to do is, if I give you a codon, you would need to be able to look it up, and then tell me what amino acid we're coded for. It is very important that you understand, and I think that's why they're trying to call it the codon chart in the lab manual. You cannot look up DNA triplets in this chart. You cannot look up anticodons to figure out how this works. You can only look up the codons that are again present in messenger RNA. That's the only thing that works in the codon chart, because mRNA is the only RNA type that actually contains the codons. Okay. From there, we've got questions that are in your lab manual. Let's see, it says use the complementary base pairing rules for DNA to fill in the chart. Now, since we're doing DNA, it's DNA to DNA. So T base pairs with A. And then you should be able to go all the way down. So you guys go ahead and practice doing that. Maybe pause the video so you can check your work to see if you're good, but I'm going to go ahead and go down and fill this in. Messily, apparently. Alright, so there's how that works. Next, this time they're giving us DNA code and they want us to transcribe it into messenger RNA. And again, notice they said transcribe. Transcribe is always DNA to RNA. So we're going to do that with step first. So T can code for A. Once again, I'm going to recommend you pause the video and go ahead and practice doing this for yourself. And then you can compare your work to what I get at the very end. Okay, so there's the messenger RNA. Then it says to use the messenger RNA strand to determine what the tRNA anticodon we're going to be. So again, we're doing complementary base pairing between mRNA and tRNA. So remember, we can't do T's once again. So A is going to go to U, G to C, like so. Then it says to use the mRNA codon and the codon chart to determine the amino acids. Now notice that they blocked off 3. Here's where what I said was very important earlier. You can only look up codons in the codon chart, which means the first codon that we're going to look up is AGG. Now here's where I'm going to back out of this, if it will let me, so that I can add this picture to where I was. Sorry guys, I should have paid attention to what I was doing earlier. Alright. That means I have to retranscribe my stuff over here. And I have to turn on my pen function. Okay, so my first codon was AGG. So once again, play that game, and then a RG, which is arginine, would be what you put off over there. UCG codes for serine. Notice that I'm completely ignoring what we would have had written over here in the tRNA box, and that's because you don't look that up in the genetic code. You only look up the codons in the genetic code. Okay. AAU, asparagine. CGU, arginine, and then AAC, ASC again. Okay, they at no point in this got us to a place for me to really talk about this with you guys, so I'm going to do it really quickly. Whenever we transcribe a gene in the messenger RNA, there's a cap and a tail that gets added to the messenger RNA, and those caps and tails kind of help the different parts hold on to the messenger RNA, or maybe those things help it get out of the nucleus. We're not really 100% sure what those things do. But what that means is there's chunks of mRNA at both ends that we don't actually want to read. So there happens to be something that's known as a start codon, which is how the ribosome knows that this is where we're actually supposed to start reading. And it's AUG, the one that codes for methionine. That's also the start codon. So the mRNA gets ignored until that start codon sequence shows up, and then the ribosome and the tRNA can lock on, and that's what sets up the reading frame, which is the set of 3 letters that we're going to be reading as we continue on through that cycle. In addition to having that, there are also, and I don't like how this code that I gave you guys says this, TER actually means stop. So UGA, UAA, and UAG, those are all stop codons that tells the ribosome, stop. You're done reading the recipe. You have cooked the dish like you're supposed to. You can ignore everything that comes after this point. So essentially what that means is the messenger RNA sort of has a capital letter at the beginning of the sentence and then a period at the end that tells the ribosome exactly where we're supposed to start and stop as we go through there. Next up, circle the correct answer for the choices as given. And we're going to assume a eukaryotic cell on this one. So DNA is located in the nucleus, mRNA is made during transcription, mRNA is made in the nucleus because again that's where the DNA is. You can't copy DNA if you're not there. mRNA leaves the nucleus to find the ribosome. mRNA or rRNA is used to carry the genetic code from DNA to the ribosome, so that's messenger RNA. RNA uses uracil instead of thymine. Amino acids make up a protein. Transcription, that's where we go from DNA to RNA, so that happens in the nucleus. TNA, that's the chef's assistant, so that gets used during cooking. Cooking is translation. tRNA uses anticodons to match up to the codons on mRNA. Proteins are made at the ribosome. We even said back in the cell chapter that the ribosome was the site of protein synthesis. tRNA just floats around out in the cytoplasm, carrying those ingredients around. Translation is cooking, so that's the process that converts mRNA to a protein. Translation happens in the cytoplasm because that's where the ribosomes are. And then, I feel like we already answered that question, but transcription converts DNA into messenger RNA. Next, fill in the chart below. DNA is double-stranded. RNA is usually single-stranded. DNA is found in the nucleus. RNA is made in the nucleus, but then it usually leaves, so we'll say it's found mostly in the cytoplasm. We'll say it messily, as always. Number of unique nucleotides. Well, DNA uses T. Or they didn't actually say number, so we'll just say T. And then the unique nucleotide in RNA is U. Name of the sugar is deoxyribose. In DNA, I can't write that small. Okay, so deoxyribose for DNA. And then just, man, I can't even write this big. O-S-E. Okay, so deoxyribose for DNA, and then just ribose for RNA. What is a mutation? It's a change, that delta means change, in the DNA sequence. How could a mutation harm an organism? It can change proteins. Now, here's the story that I will give you to go along with this. We're going to come back to this little thing that we had done earlier. So in this original strand of DNA that we had, that was the sequence. Let's say that there were a mutation, and that mutation changed that to an A. What would that mean? It means that when the cell carries out the process of transcription and then translation, we've got this new letter that we have to deal with. So I'm going to change my colors just so that you guys can see the differences kind of as we go through there. I have to remember I've got to do that first, and then this. Okay, so if we change to an A, that means this would be a U. That means our new codon would be AUG. Where our original ingredient told us we were supposed to add arginine, AUG doesn't code for that. Instead, it codes for MET. So using my cooking analogy and my lemon bars that I made earlier in the week, let's say that the arginine was the lemon, but I changed the lemon out, and instead MET is for tomatoes, and so I made tomato bars instead of lemon bars. Is that the same thing? Would you guys want to eat some tomato bars that I made? Probably not. So ordinarily, if you change an ingredient that was used to make a protein, you change the dish that gets made, and now maybe that dish can't be used like it's supposed to anymore. The most common case that usually gets talked about at this point in high school biology and college classes for that matter is sickle cell anemia. People that have African ancestry are more likely to have a mutation in their hemoglobin gene where one of the letters gets substituted for a different letter, exactly like we did right here. This is called a base substitution, where one letter gets changed for another. As a result, their hemoglobin contains one bad amino acid, and by changing that one amino acid, they change their hemoglobin enough that it can't do its job anymore. When it starts to unload oxygen, the bad hemoglobin actually changes the shape of their red blood cells, and it makes them sickle-shaped, and it makes them firmer. And as a result, they can't bend through capillaries like they're supposed to, and so they start to dam up or block the capillaries, and that causes what's known as ischemia, which means tissue starts to die because blood is not flowing to tissues anymore. So all because of that one mutation. It changes the whole dish that got cooked, and now a person can't do something that they used to be able to do. Now, sometimes mutations are actually beneficial. There are mutations in Sherpas, who are people that live in the Himalayan mountains, that allow them to make better hemoglobin and more hemoglobin that has a stronger binding affinity for oxygen, so that's why they're so good at traipsing up the Himalayan mountains and helping people get up those mountains. It's because their hemoglobin is actually better than my hemoglobin or people of African descent's hemoglobin. And so mutations can be harmful, they can be beneficial, or, and I'm going to actually do this one with you guys, so let me see if AGG would have worked for that. Alright, so now I'm going to play around with a different one. So TTG, we're going to play here, but I have to think in my head for just a second, so bear with me. I need this to be that. Okay, so we're going to change the G to an A. As a result of changing the G to an A, this C becomes a U. And so what had been AAC is now AAU. Go away, stupid screen. AAU still codes for that same ASN. That is what's known as a silent mutation, where we changed the DNA, but by changing the DNA, it still brought the same ingredient to the dish, so it didn't actually change anything inside the cells. Silent mutations are actually very common in that third place within a triplet or a codon. Alright, so that's how mutations can actually harm an organism or not. It is important that you understand that mutations may not actually harm an organism. They might harm the organism or they might be beneficial. So that, I believe, concludes your DNA chapter. Good luck on the quiz.